This is a production of Cornell University.
Welcome everybody to the fall seminar series for soil and crop sciences. We have a strong lineup of presentations, including some panels later on, and we have a lot of student presenters for this semester. So we're delighted to begin today with Olenka, who's in Elena's lab, who will be, well, Olenka, I'll let you explain the title of your talk, give the title of your talk. Couple rules of the road for the day. If you have a question while Olenka is speaking, please type it into the chat and I or Olenka will read it and answer it at the end of the session. And then time permitting, we'll also open the floor to questions at that point. So without any further ado, welcome. Thank you a lot for attending. And Elena, over to you to introduce Olenka.
Thank you, Andy. So, well, hello everyone. It's unusual seminar series, unusual semester. Well, first of all, I would like to take this opportunity to introduce our new PhD students. Unfortunately, only two can be here virtually, and it's Kiera Crowley. Maybe Kiera wave or say hi so that all
hello.
People. Yeah.
A lot of people here, well, some people here know me already, but
right.
I'm sure there's some people I don't know too, but I'm a new PhD student with Andy McDonald.
Right, and Kiera was an MS student with
Matt Ryan.
With Matt Ryan.
That's right, yeah, so I came back.
And Sophie Westbrook. Sophie, please say
hello.
Hi.
Good to meet you all. I'm a new MS PhD student with Tony DiTomaso.
Unfortunately, two PhD students, Jiameng Lai and Zhenqi Luo, they cannot be with us because it's actually midnight or past midnight in China. So Zhenqi and Jiameng, they are students with Ying San. And Jiameng, she's actually enrolled and taking classes. She'll join us probably at the end of the semester, and Zhenqi will join us hopefully in the spring. And so, yeah, so welcome to the family. And now I would like to introduce Olenka, although I think for many of our PhD students, Olenka doesn't need a lot of introduction because she's very active in our graduate student association. So she served as a vice president and last year was a president. And I think now she's ready to pass the torch to a new generation. So I'm encouraging all the students to be very active in this GSA, in our GSA. So Olenka originally is from Ukraine, from the same city that I am, from Lviv, which is, you know, in a western border with Poland. And that's where she was, born, raised, and that's where she obtained her bachelor degree. She graduated from the national. Before it was Lviv National University, now it's National University of Lviv. There's changes. I actually graduated from the same university too. So it's a pleasure for me to host Olenka. So Olenka came to US in 2015, and she initially joined my lab as a technical assistant. And then she decided she wanted to continue. She actually switched because she did her bachelor's are in genetics and biotechnology. And back in Ukraine, she was working with microorganisms and trying to engineer microbes that can serve as factories for antibiotics. And so she switched to plants, and she found that she has to be very patient with plants because it's not 12 hours of, she cannot get results in overnight. So she has to wait and wait and wait, and then wait some more. And so, I think in 2017, Olenka started her PhD, MS PhD, and now she is post her A exam. Actually, today is her pre-A, post-A seminar. She successfully passed her A exam in June. And she is already co-authored on 2 publications and is starting her research project that will lead to her first author publication, hopefully within the next several years. And Olenka's seminar title is quite long, but briefly what she's going to talk today about is about the transcription regulators which are involved in the regulation of copper and iron homeostasis in the global crop wheat and Brachypodium. And I think with this, I'll pass it to Olenka.
Thank you, Anna. Hi, everyone. Welcome to the seminar series. It's quite unusual to not be speaking to a room full of people, so I'm just speaking to many rooms with each person in them. I'm going to start with explaining what my talk's about, and if you have any questions, feel free to ask them. So the topic of my presentation is establishing the role of CITFL-Like genes in the regulation of copper and iron homeostasis and reproduction in wheat and its model Brachypodium. So here's, sorry, need to switch it. Here's a short outline of what I will be talking about. So I will be talking about a bit micronutrient deficiencies and how they're connected to food security, about roles of iron and copper, about the VHLH family of transcription factors that are involved in plant mineral homeostasis, and how they're connected to crops. And obviously, I will give you some idea of what work I'm currently doing in the lab. So why do we need to be interested in micronutrients such as copper and iron, and what is their role in food security? To answer this question, let's figure out what actually is food security, or more basically, one of the problems is food insecurity. So food insecurity is defined as the situation where people lack secure access to sufficient amounts of safe and nutritious food for normal growth and development and an active, healthy life. And about 800 million individuals across the world are considered undernourished. And even though UN had set this global goal to end hunger by 2030, we're currently pretty far from reaching that target, as some of the countries currently have food deficit that are below 200 kilocalories per person per day. However, we have to realize that calorie deficit is not the only indicator of undernourishment, and this leads me to talking about micronutrient deficiencies in humans, since a term called hidden hunger is actually a form of undernutrition that occurs when intake and absorption of vitamins and minerals are too low to sustain good health and development. Obviously, deficiencies of micronutrients will show some symptoms in people, and iron deficiency is most well known, I suppose, as anemia. So symptoms of anemia, here you can see on the map, are usually among preschool age children. You can see it right away, and the symptoms are usually extreme fatigue, weakness, cold hands and feet, and pale skin. However, if you look at copper deficiency symptoms, they're pretty similar. The reason for that is actually copper is pretty important for iron absorption. So it's really connected together in humans. For them to be more healthy, they really do need these micronutrients. And as you can see from this map, particularly acute cases of anemia are in the countries where people are less fortunate with resources and have to rely on plant-based diet only. So one of the ways to resolve this issue is biofortification. Biofortification is a process that increases bioavailability of and the concentration of nutrients in crops, both by conventional plant breeding efforts and through biotechnology. Since fortification of food can be performed in some countries where it can be added globally, for example, we add iodine to salt. However, biofortification will probably benefit more places that are more rural, and that way you already will have the plant that is able to get all of its nutrients and increase the amount of nutritional value right away. Since plants take their nutrients from the soil, if they actually lack some of them, you would be able to see it on the plant right away. So symptoms of some of them are pretty acute. For example, for the symptom of iron deficiency, you can see that plants have this intervilleinal chlorosis, of young leaves, which is like a huge yellow flag, I would say, not the red flag, to show that something's wrong with your plant. Among other symptoms are obviously swelling of root tips, and in acute cases, even full crop failure. Symptoms of copper deficiency, however, can be a bit more subtle. It's again, decreased growth rate and chlorosis. However, another thing that you can see is impaired lignification of cell walls, which results in curling of the leaves and weaker stems and lodging. Another thing is flower abortion, and as a result, a lower seed and grain set. So why the nutrient deficiencies occur? Well, one of the reasons would be that micronutrient availability really fluctuates depending on the soils they're in, and more than 30% of world's arable land are considered to be iron and copper deficient, since their pH are alkaline and neutral, which makes these micronutrients unavailable. Even though micronutrients is important for our health, we have to remember that in high amounts, they can be toxic, and therefore plants need to know how to regulate their uptake and transport using proteins called transcription factors, or TFs, as I will be referring to them later. So as I was mentioning, iron causes such an issue as anemia, so it has been studied more extensively. One of the most important transporters that help uptake iron in the plants is called IRT1, so I just want you to remember this name. And the processes of iron uptake really vary in different plants. At first, they were really strictly divided to two strategies, such as reduction strategy and chelation strategy. And reduction strategy was considered to only be used by dicots and non-grass monocots, so they would reduce iron 3 to iron 2 to take it up. And chelation strategy was more preferable for grasses, where they extrude chelates such as phytosideriphores, and then iron 3 gets chelated and taken up. However, the more iron was studied, the more we knew about it, and actually this division came to be shortened, and you could see that actually the uptake of iron 2 by IRT1 can happen in plants from both strategies. All of these processes, however, are controlled by the transcription factors, and specifically by the BHLH family of transcription factors. So from here, I want you to remember BHLH family of transcription factors and the IRT1. Moving on, we will talk about that copper, unlike iron, hasn't been studied that much. For a long time, the only transcription factor that regulated copper deficiency symptoms that has been known to everyone was actually SPL7. And until recently, that was the only real big way to know of regulation of copper homeostasis is to look at SPL7. However, a student in our lab, Japan, you probably remember her, actually discovered that there's another transcription factor that regulates copper homeostasis, and it's called CITF1. So here you can see that she worked on Arabidopsis plants, which is a model for dicot plants. And what I want you to pay attention to here is, here you can see the sleeks that have grains inside of them. So here you can see the wild type sleeks that is filled with grains. Then you could look at CITF1 mutant and SPL7 mutant that generally still have grains. However, if you look at the double mutant of CITF1 and SPL7, the plant that lacks both of these transcription factors, you can see that there are no grains inside because it's turned out to be 90% infertile. And that is something that was really interesting to us. And we decided to see, well, how do we know that it's actually copper that we have to look at? For this, we employed this method that's called synchrotron X-ray fluorescent microscopy. And my lab mate, Rujan, was working at synchrotron source here at Cornell, which is actually across the field from Bradfield. And what was found is you can actually visualize all the minerals you need in the tissues of plants using this technique. And here you can see the density of copper in the flowers of Arabidopsis. So here you can see wild type plants that actually have plenty of copper in their flowers, and specifically in the anthers. Those warmer colors show a higher density. Later, when we look at different mutants, you can see that copper density actually goes down. It's lower in CITF1 mutant, it's even lower in SPL7 mutant, and it's basically not there in the double mutant. So it's very interesting. So since CITF1 seems to play an important role, we decided to look at its place among other BHLH transcription factors, and did a phylogenetic tree of more than 160 BHLH genes. And this is the close-up of that big tree. And what I want you to see is, here is CITF1, and it's controlling copper homeostasis. However, it is in the same clade as some of the transcription factors that are involved in iron homeostasis. So again, copper and iron come together. So all of that was actually studied in Arabidopsis and dicots. However, how are copper transport processes regulated in grasses that include cover crops such as wheat, rice, and corn is not well studied. That's why it's of interest to me. So for these studies, we've chosen to concentrate on wheat as one of the most important crops, and its model Brachypodium. The reason I use Brachypodium as a model for wheat is because its genome is less complex. It's smaller, it's fully sequenced, the plant has shorter life cycle, smaller stature, and it's more amenable to genetic transformation. So it's really cool. It's considered to be a model, but we decided to make sure that our results are reproducible. So we decided to compare the response to copper of Brachypodium compared to wheat and see if these results are actually reproducible. So here you can see that we've grown both wheat and Brachypodium on different concentrations of copper from 0 to 500 nanomolar. And what we did, we evaluated the amount of flowers that are being produced and amount of grains that are being produced depending on concentration of copper the plants were grown in. And something I want you to pay attention to is that, for example, if you look at plants grown at 15 nanomolar copper, if you look at the number of flowers, yeah, they're bigger than with like 0 copper, for example. There's a larger amount of flowers over here or over here. So here you can see wheat flowers and Brachypodium flowers. However, if you want to look at the grains, the number of grains goes down, which indicates that copper is pretty important for grain yield in both in wheat and Brachypodium. Since like you would be going down the field, for example, and you see beautiful flowers and there are a lot of flowers in the field and you're like, it seems I'll get a nice yield this year. However, as you can see, the amount of flowers doesn't necessarily mean that you'll get grain yield. And so this little test showed us that indeed Brachypodium shows results that are comparable to results in wheat. So obviously I can use it as a model for wheat studies in my later studies. So what I did next is I decided to look if there are any CITFL-like genes in wheat and Brachypodium, and found that indeed CITF1 does have several homologs both in wheat and Brachypodium, and I designated them CITFL from CITFL-like 1, 2, and 3 for wheat, and CITFL 1, 2, and 3 in Brachypodium. So this leads me to my specific objectives for my PhD studies, which are establish the role of CITFLs in copper and iron homeostasis and reproduction in Brachypodium, as well as in wheat, identify genes regulated by CITFLs in wheat and Brachypodium, establish the relationship of CITFLs to the SPL7 pathway in regards to copper transport processes and reproduction, and analyze protein-protein interaction of CITFL transcription factors. So I started with looking at the expression levels of my genes in different tissues of Brachypodium, and saw that they're highly expressed in stems, flag leaves for CITFLs 1 and 2, and in roots for CITFL 3. So basically those are the tissues you would use, I would use for later expression studies, which I did. And if you look here, I looked at the expression of CITFL 1, 2, and 3 under copper and iron deficiency. And as you can see, here is a full difference of ratio. So ratio between the copper or iron deficiency and control conditions. And you can see that all 3 of them are actually upregulated under deficiency conditions, which is pretty cool. It's something that I wanted to see, and to test the biological relevance of this increased expression, and to see if is it important enough or not. I need to make mutants of these genes. So I decided to use the CRISPR Cas9 genome editing system And just to remind you how it works, so the components of this editing system are genomic DNA that you're going to be editing, guide RNA that you actually design to lead the system to edit in specific place you want it to edit, and a nuclease, Cas9, which actually does the cutting. So the Cas9 forms the complex with guide RNAs, and the guide RNA finds and attaches to the sequence in genomic DNA, the one that you planned for it, after which the complex cuts the sequence, and after that, the genomic DNA is being repaired and the mutations are introduced in the process of preparation. So I will show you on an example of how I made my mutant from CITFL-1. So here you can see a genomic DNA sequence of CITFL-1, and I generated guide RNAs. The reason I generated more than one guide RNA is because I wanted to make a deletion out of this gene. So if you make 2 guide RNAs, you you can have this chunk of around 500 base pairs to be cut out of your gene. So the reason we have several guide RNAs is because we're trying different combinations to see which one of them works out. So for example, for me, the one that worked out was combination 1+3, and my expected edited size would be around 1,000 base pairs. So that's what happened. The nuclease is cut in the process, and this whole chunk of DNA was cut out, after which it was repaired. And here you can see the end result. However, this is just what's actually happening during the editing, but it takes so much more time and effort to actually get the mutant, since transformation of Brachypodium is a bit of a long process. So first you have to start with dissecting the embryos of Brachypodium, then initiated callus, and after co-culturing colors with the bacteria that has your construct, you have to select it on appropriate antibiotics and then regenerate it. As you can see, all of this takes a lot of time and effort until you can finally have your mutant plants. However, the fact that you got the plants doesn't necessarily mean that all the editing happened. For this, we have to do some checking, and the way we do it is each of the plants we regenerated, we collect their tissues, extract their genomic DNA, and run a PCR. And here you could see the expected results of DNA electrophoresis. So if editing didn't happen, the result will be similar to the wild type. So it will be around 1.5 kb, a bend on the electrophoresis gel. However, if your editing did happen, in my case, it's an editing of 1,000 base pairs. So here on the 0.5 kb, you can see a bend, and that will mean you have a homozygous mutant. However, if you see both bends on the gel, that means you got a heterozygous mutant. So in order to get a homozygous mutant, you will need to get later generations of plants. Another thing to look for is whether Cas9 nuclease is still inside the plant. As you know, it's very useful because it does all the editing. However, you kind of wanted to edit and then go away and not to keep doing the work all the time, since you need to know actually what type of editing happened, and you need to know it's not happening anymore. So for that, you also perform PCR that detects for the Cas9, and mutants that still have Cas9 have the band of approximately 0.7 kb. However, both mutants that already don't have the Cas9 and the wild type won't have any bands. So here I will show you the obtained results, and as you can see, I've obtained 4 mutant lines. 3 of them are homozygous and 1 of them is heterozygous. However, when I looked for the Cas9, I saw that only line number 1 was Cas9 free, and this is the one that I'm using in my further studies. So after I found that my mutant was obtained, I needed to sequence it to see what deletion I got, what is the size, where it is. So it showed me that I got 1,113 base pairs deletion. So approximately the size I was planning for. And what you do later is you have to look if the mutant shows any phenotype. This is what I did. So I grew it on different media, that is control media with all of the necessary nutrients, iron deficient media, and copper deficient media. And as you can see from my preliminary data, that even in control conditions, the mutant has smaller stature, so it already shows some phenotype. And under deficiency conditions, it seems to have a lower amount of leaves. So this is my preliminary data. I just finally got my mutant, so that's something I will keep analyzing in the future. Another thing that I wanted to look at is whether CITF1 is connected to SPL7 in the grasses, since they are connected in the dicots. So my lab mate, Huajin Zhang, he was working on the SPL7 homolog in Brachypodium, and he found that it is required for copper uptake, delivery to flag leaves, florets, and fertility. So here you can again see the results of X-ray fluorescent microscopy studies. And these are the flowers of Brachypodium. And as you can see, in comparison, the mutant doesn't have a lot of copper in the flowers. So I decided to use his mutant to look at the expression of my CATFL genes and see if there's a connection. So what I expect is if the transcription factor, which is SPL7 in this case, if it actually somehow regulates your gene of interest, we will see changes in the gene expression of mutant plants. However, if it doesn't, there will be no change in gene expression. So that's what I did. I looked at both wild type and SPL7 mutant and in different tissues, and all 3 CATFL genes were actually regulated by SPL7 in Brachypodium. However, for example, CATF1 and 2 were downregulated in roots and young leaves. However, CITFL3, for example, was upregulated in the old leaves. So changes in regulation show me that indeed there's connection between CITFLs and SPL7. One of the other things I did is I used bioinformatic approach to look at other potential interactors for my CITFL genes. And I used a gene regulatory network that predicts the interactions for my genes of interest and I found that most of them are connected to iron homeostasis. So I don't want you to remember all of these names. There are a lot of them, and it's usually an online software that predicts all of it, and you just pick something that's of interest to you at the moment. So what I want you to pay attention is to IRT1 that actually has a connection with CITFL1. And I will remind you that IRT1 is actually an iron uptake transporter, as I already mentioned. And another thing I wanted to remind you is that CITF1 in Arabidopsis is in the same clade as transcription factors that are involved in iron homeostasis. So what I decided to do is to see whether IRT1 is possibly regulated by CITFL1 in Brachypodium. So again, the same type of scheme. You can see here, if transcription factor regulates gene of interest, I will see changes in expression of mutant plants. If it doesn't, I won't see any changes in expression. And indeed, I actually saw changes in expression for IRT1 in my CHTFL1 mutant. And that means that possibly that CHTFL1 actually regulates IRT1 and potentially other iron-related transporters. Another thing I wanted to look at is to see how my CHTFL transcription factors act. So one of the peculiar things about the BHLH family of transcription factors is they cannot act alone. They have to be paired up or dimerized to act. And the way they do it is they have several domains on them, and part of them are responsible for protein-protein interaction. So this is the part that would be responsible for protein-protein interaction. So one of the BHLH family members will connect their protein-protein interaction part to the same part in a partner that is another member of the BHLH family of transcription factors. And if they connect together, as you see over here, these proteins are connected, and then they kind of assemble in a way that they both can interact with DNA and attach to it. So one of the things you could do, and one of the things I did, is look at possible interactors among other BHLH family members. And here you can see yeast studies that were used for it. So basically, I decided to see if my Brachypodium CATFL 1, 2, and 3 are interacting or not. So the way you pair them up, you can pair them up as a CTFL1 with CTFL1. Maybe it needs 2 of them to act, or CTFL1 with CTFL2. So either type of combinations, and I tried most of them. However, the only one that actually showed an interaction was CTFL1 with CTFL1. So 2 proteins need to interact, and if interaction happens over here, you will see the growth on the selection media, on the quadruple dropout selection media, or on the selection media that actually shows the blue color. So if the blue color is there, there is an interaction, and if it's not, there is no interaction. And indeed, these results show that there is an interaction between CATFL1 and CATFL1. And that is pretty cool, and I hope to also work on looking for some other BHLH members that might interact with my CATFLs. So in conclusion, I want you to remember several things, which are CITFL 1, 2, and 3 in Brachypodium are upregulated by copper and iron deficiency. CITFL1 mutant in Brachypodium that was generated using CRISPR Cas9 gene editing system shows that the preliminary study showed that the mutant grows slower and actually shows sensitivity to iron and copper deficiencies. I also found that IRT1 is downregulated in CITFL1 mutant, which suggests that CITFL1 transcription factor regulates iron uptake transporter IRT1. I also found that CITFLs are regulated by SPL7, which shows insight into copper homeostasis network. And I also found that CITFL1 interacts with itself in used-to hybrid studies. So there's still some work left to do for me with establishing the role of CITFL1 in copper and iron homeostasis and reproduction. I plan to test the sensitivity of the mutant to copper and iron deficiency, test the ability of it to accumulate copper and iron in its tissues, test the fertility of the mutant and compare it to the wild type plants. I would also want to establish the relationship between CITFL1 mutants to SPL7 pathway in relation to copper transport processes. And for this, I would like to generate a double mutant between CITFL1 and SPL7 and look at the phenotype of this double mutant and analyze its flower development and morphology. Another thing I want to do is identify more genes regulated by CITFL1, and for this I will use deep transcriptome analysis, RNA-seq, to identify the genes that are regulated by CITFL1 in different tissues under copper and iron deficiency conditions. And I will also like to use yeast studies to see whether CITFL1 is binding to IRT1. To do these studies, I actually got the Schmittau-Novak Integrative Plant Science small grant for this year. So yay, I've got the funding to do what I want to do. So I would like to show you that out of the specific objectives of my PhD work, I talked about 4 of them. However, the establishment of the role of CITFLs in copper homeostasis and reproduction in wheat is a more complicated process since I'm not able to make a mutant on my own. We're gonna collaborate with plant transformation facility with Matthew Willman there, and they will be taking care of our mutants. And once I get mutants from wheat, I will repeat all of the work that I've done with CITFL1 and still plan to do with CITFL 2 and 3 in Brachypodium. I will repeat the same work in wheat once plant transformation facility gets me those mutants. And with that, I would like to thank all of my lab members. Thank you to my committee and our collaborators and funding. And with that, I would be happy to take any questions.
Thank you, Olenka.
Yeah,
So-
very well done, Olenka.
Yep, well done. So questions? Since I didn't see any questions in chat, so just read.
Oh, someone raised a hand.
Yeah.
Oh, hi.
I'm Tim Sutter here. I have a question. So the work that you presented of your own and also of your colleagues is showing a particular importance of copper and iron in the reproductive organs. Can you explain why the reproductive organs have this special need for copper and iron?
So as you can see, for example, sorry, I'll click real quick. Here. So as you can see here, we looked at different concentrations of copper and like how different plants react to them. And as we've shown in XRF studies that in copper regulation mutants, copper is not there. And as you might have noticed, I showed the anthers and as part of the reproductive system. So one of the interesting things is anther dehiscence. So apparently if you don't have enough copper, the anthers won't dehisce and pollen won't be viable, since copper takes part in a lot of processes such as lignification, which is important for anther dehiscence, and is also taking part in so many cell processes, such as electron transport processes. So basically, you get the energy in the cell. However, if there isn't enough copper, some of these processes are not happening, so pollen is not viable, and therefore fertility goes down and you cannot get the yield.
Okay, thank you.
Johannes has his hand up.
Hi, yeah, thank you very much. Looks great. I had a question to the synchrotron studies. One thing that we had always problem with is if we compare different measurements, the intensity is very difficult to compare. Are you putting all the samples into the same view to be able to compare them? Or are there some ways to normalize the intensity? And the second question was, is there a way to also say something about the copper forms that would be instructive for you in addition to the spatial distribution? Is there any hidden opportunity there? Would be interested to know. Thanks.
Thank you for your question. So as for XRF studies, the more professional person to ask in our lab is Rujan. She is actually the one who's using all of this. However, I will try to answer your question better. Yes, these concentrations are comparable. As for distinguishing between different copper forms, I believe we haven't done that. However, Olenka probably knows more about this. Yeah.
So Rujan, actually she's here, so she can add if she wants, if I miss something. So yes, this is very good, excellent point, Johannes. Indeed, in order to be able to compare, you really have, what we do, we actually put our samples side by side. So what are you seeing here? It was scanned at once. So it is comparable. The difficulty that we are having, although in this case it's nearly all enough or nothing, right? If you look at the double mutant versus each of the mutants, one of the issues that we were considering before is the thickness of the sample, right? Because if the plant tissue is thinner because of it's just smaller, it's just because of copper deficiency, etc., then you will be assuming that it's less copper. But so that's a difficulty. So we overcame this difficulty with like an excellent tool that has been developed here at CHES. It's called confocal synchronous X-ray fluorescent microscopy. So basically, what this tool allows you is to take optical section and actually measure intensities in a defined thickness. So this is not what it is currently, but we've done it, and we know that we can do it. And we've done it, and we compare. So basically, results are reproducible, and we can do it like at 5 microns. I mean, they can go lower, but 5 microns, that's the reasonable resolution. With copper speciation is also very, I mean, it's something that we would be interested in, right? Copper, as you know, and probably many of you appreciate, is, it can be toxic. Actually, copper is considered to be more toxic than iron in a cell, and copper doesn't hang by itself. Like you won't, if it's free in a free ionic state, then it's toxic. And so even movement of copper in the cell, it has to be controlled. And there are chaperones that move copper to enzymes, etc. And there are a whole bunch of ligands that help to move copper. And so, of course, to know how copper, in which form, in which state, is it copper 1 or copper 2? Is it with acids? Is it with nicotinamide? We want to know. And we can also do it, well, we can do it at chess once it's, you can do Zanes, XAFs, etc. So there are opportunities there. Yeah. Rujan, do you want to add? I know that Rujan is somewhere around, but I don't know.
No. I think you already answered the question very well. So, yeah.
So Rujan is our XRF guru. So if we, like she started this work at chess, and now we're moving to Brookhaven. So kind of, right? So that's her baby.
Yeah.
Okay. Other questions? All right. So I think we're just on time. It's 1:31. So let's thank Olenka for a nice presentation, and good luck with PhD degree and other studies. Thank you. I clap. I know. Yeah.
Thank you.
Yeah.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.